Welcome everyone. This is the DNA testing in practice, module 4. So we're going to focus on methylation, folate and B vitamin metabolism, a topic that is very close to my heart and that I've been working in this particular area of methylation for a very long time, since the early 2000s. The topics covered today are the methylation cycle. So I'm going to explain exactly what methylation is. What is a methyl group? What does methylation mean? But I'm not going to spend a long time on that explanation. I think many of you have been working in this area for some time and you are willing to go a little deeper. So I'm going to go into quite a lot of depth around folate metabolism, which is absolutely essential for proper methylation. The reason we're going to go in deep with folate is because the questions that I get most frequently are, if you've got this genetic SNP, what does that mean for this particular supplement? Or can you compare folic acid with 5-methyl tetrahydrofolate? Or what's my opinion? So I'm going to give you the evidence. I'm going to try to be as objective as possible so that you, hopefully at the end of this module, feel a lot more in control and educated and have that evidence to make those decisions for your patients and your clients. We'll also, of course, talk about the homocysteine and methionine metabolic pathway. We'll talk about B12. Know that B12 is a huge topic as well. I'll address the different forms of B12, but you'll get more of that in the neurotransmitter module as well. A lot of these things cross over. So what I'll try to do in each module is have a sort of a focus, and I'll touch on the important things that are related, but then in the coming modules we'll expand. So you'll continue to build as these modules go. The learning objectives for this module are to describe the methylation cycle, understand the important SNPs or the genetic variations, to examine the enzyme cofactors. So of course we know that genetic variations can influence the activity of these enzymes, but they need those cofactors, whether it's B2 or B6 or B12. To identify important conditions that may be affected by imbalances in methylation. So these are the health and disease outcomes that we know are influenced by methylation. To recognise the numerous factors known to balance or alter methylation, in addition to the SNPs and folate and B vitamins, and to build effective clinical strategies. You will get more of that when we really get into the case study as well. So what is methylation? So methylation actually refers to the addition of a methyl group, and a methyl group is simply a carbon and 3 hydrogens. So often a methyl group is even called a 1-carbon group. So the process of methylation is literally the adding of these methyl groups onto compounds. But often when we say methylation, what we really mean is this methylation cycle. So we're talking about the folate, the B vitamins, the genes like MTHFR that influence methylation and actually go on to make SAMe or S-adenylyl methionine, that is the universal methyl donor or methyl donor that affects methylation throughout the body. So this is a simplified version of the methylation cycle. It's also called the 1-carbon metabolic pathway, or people often refer to it as the homocysteine or folate homocysteine metabolic pathway. It's all the same thing. I'll step you through it briefly, but know that we're going to go through this in detail, so we'll break this down. So on the right, you can see the folate pathway, and here you can clearly see the different forms of folate and where they enter the pathway, or at what points they enter this folate cycle. At the top, we have folic acid. So folic acid has to go through or be metabolized by dihydrofolate reductase to form tetrahydrofolate. Now that's a little simplified, but we will talk about it in more detail in the coming slides. We can see there's folinic acid that comes in as 5,10-methylene tetrahydrofolate. So this is an important form of folate that is needed to build and make the nucleotides. These are essential for DNA synthesis and DNA repair. And then we have 5-methyl tetrahydrofolate down the bottom. So 5-methyl tetrahydrofolate is what donates a methyl group to homocysteine. So moving over to the homocysteine methionine side, that methyl group is donated to homocysteine, and then via methionine synthase or MTR, that methyl group is added onto homocysteine, forming methionine, We know that we can also get methionine obviously from the diet. This is an essential amino acid needed for protein synthesis, but also for making S-adenosyl methionine. So S-adenosyl methionine, many of you call SAMe, and that goes on to provide all of those methyl groups for these methylation reactions throughout the body, whether that is metabolizing hormones or breaking down neurotransmitters or helping with energy production. So we'll go through these genes and we'll break this pathway up to help you understand that a little deeper. I'll just note you can see the transsulfuration pathway down the bottom. We'll focus on this in the detox module, or we'll talk about CBS in module 6 in detoxification, and we'll also talk about these genes in relation to B12 that are on this slide. So why is methylation important? I've already mentioned a few things. We know that methylation is essential for gene expression. It's one of the major or most well-known epigenetic marks. So this is important for regulating what genes are going to be expressed or turned on. We know that it's important for DNA synthesis and repair. So I mentioned in the previous slide 5,10-methylene tetrahydrofolate, that form of folate that sits above 5,5-methyl. So that's essential for donating a methyl group onto uracil to make thymine, one of the 4 bases of DNA. So absolutely essential when our DNA is replicating, when we're making new cells. It's also important in detoxification. One example is breaking down and trying to metabolize and excrete arsenic. We'll talk about this in the next module. We know that it's important for helping to metabolize catechol estrogens, so balancing our estrogen levels. It's also involved in the breakdown of neurotransmitters and energy production. So let's get into folate. So the word folate is a generic term that actually represents all forms of vitamin B9. Folates differ in their 1-carbon substituent. This means the methyl groups. Remember, 1 carbon is a methyl group. Folates also differ in the number of polyglutamate residues, and I'll explain what this means. And folates also differ in their oxidation state. So folic acid is oxidized, whereas the natural food folates are the reduced form. The natural forms of folate include the tetrahydrofolates, and we usually see this written as THF or THFs, and they are structurally related, as you'll see. The major form of folate that is circulating in our blood is 5-methyl tetrahydrofolate, and folic acid, or pteromonoglutamic acid, or often called pteroglutamic acid, is the oxidized and synthetic form that we usually see in vitamin supplements and fortified foods. So here we can see the chemical structures. I definitely do not expect you to remember these. This is not something that you're going to be questioned on, but the reason I'm showing you is I want to show you these slight changes to help you understand the differences in folate. So this slide shows the chemical structures of folates. You don't need to memorise these. The point of showing you these is to highlight the differences in these particular chemical structures and these various forms of folate. So we can see in the top with pteroglutamic acid or folic acid, it has 2 double bonds. For this then to be converted to dihydrofolate, which actually can be made naturally, one of those double bonds has to be removed. Then when we form tetrahydrofolate, now this is an active form of folate. Tetrahydrofolate is what can pick up these methyl groups and pass them around that methylation cycle. It actually loses both of those double bonds. And in addition to not having the double bonds, what we can see here at the end is you'll notice there's a little N. So there's these glutamic acid residues or glutamate groups. So there are monoglutamate forms of folate, and actually folic acid is a monoglutamate. But when we eat folate, when we eat the food folates, they're actually in a polyglutamate form, meaning they've got a number of glutamic acid residues. So when you see that little N there, we're saying there's a number of glutamic acid residues. Then we have 5-methyl tetrahydrofolate. So this has a methyl group stuck onto it. And here we've got 10-formyl tetrahydrofolate, which actually has a different group added to it. So there's slight changes, but these slight changes affect where these folates enter the pathway, what enzymes they will be metabolized by, and the function of these. So why is folate so important? We've obviously talked about all the functions of methylation, and folate is critical for methylation. Folate is the substrate that picks up these methyl groups. It's needed for the synthesis of nucleotides, as we've mentioned, therefore cell growth, and it's also very important for metabolising or keeping homocysteine in a healthy and optimal range. Folate deficiency is linked with increased risk for DNA damage, and this is an underlying trigger or factor for many diseases, cancers, neurological diseases, etc. We know that folate deficiency can affect methylation, so it can cause hypomethylation if there's not enough methyl groups or imbalances in that methylation pattern. And it can also cause hyperhomocysteine, so high levels of homocysteine. We know that hyperhomocystinemia or high homocysteine can cause endothelial cell dysfunction. This obviously affects the blood vessels. This is linked with cardiovascular disease, linked with pregnancy complications. When you think the placenta is a highly vascular organ, the brain, all the blood vessels going to the brain. So homocysteine is associated with a number of diseases and health disorders. So folate cannot actually be made by humans. So we need to get it from the diet or our nutrient intake. In saying that, we do know that some of the bacteria in our gut can produce folate. However, it's only a small amount and really is not enough. We need to get it from the diet. The dietary folates, as I mentioned before, usually come in the conjugated state with this polyglutamate chain. So they have these glutamate groups added to it. But then when we eat the folates, what happens is these glutamic acid residues or glutamate groups actually have to come off, because for folate to be absorbed and get across that cell membrane, it needs to be in the monoglutamate form. So the food form is the polyglutamate. We then need to remove these glutamic acid residues, or also known as glutamate groups, so that we can absorb them. Most of this is done in the mucosa of the small intestine, and some can happen in the liver, but just a small amount of these conversions happen in the liver. This diagram is to try to help you understand that. So here we can see that we're eating folate, it's in the polyglutamate form. We have specific enzymes that actually cleave or cut off these glutamate groups so that we have folate in that monoglutamate form, and then it can be absorbed across the cell membrane and into the cells. If we want the folate to stay in the cell, because it's going to bind to a particular enzyme, or we need to use that folate to make thiamine or for DNA synthesis, those glutamate residues are actually added back onto the folate. That keeps it in the cell. If the folate has to go to the liver or to red blood cells, or there's a developing baby, it'll stay in the monoglutamate form so that it can cross the cell membrane. So when you're measuring folate in the blood, you're looking at the monoglutamate form. And most of the folate in our blood is actually in that 5-methyl tetrahydrofolate form. So why am I going on and on and on about monoglutamate and polyglutamate? You're like, really? I've had enough of this. It's so that you actually understand some of these differences in folic acid and the natural forms. Because if you are like me, 15 years ago, I remember learning that folic acid is actually better absorbed than food folates. And folic acid is said to have a bioavailability of about 100% if someone takes it on an empty stomach. So that's because it's in the monoglutamate form. It crosses the cell membrane. That's why it's absorbed better. But what I didn't learn 15 years ago, well, actually I did, but it wasn't explained like this, is that yes, folic acid is absorbed better. It gets across the cell membrane, but it's not actually active. If it doesn't go through dihydrofolate reductase to form dihydrofolate, and then go through dihydrofolate reductase again, so it has to go through the same enzyme twice to form tetrahydrofolate, it's not actually functional. So it's absorbed better, but it needs to be reduced. It needs to go through multiple enzyme steps before it's actually active. And then the other thing to note is the polyglutamate forms of folate are actually more efficient substrates for the folate-dependent enzymes. So when we're talking about that methylation cycle and all of these reactions that are going on, it's actually the polyglutamate forms, because the polyglutamates are stuck back onto the folate, that are going to be used in the cells. The bioavailability of food folates is dependent on the glutamic acid residues, how many are there, the dietary and non-dietary factors that facilitate or inhibit folate absorption, and it's estimated that about 50% of food folates are absorbed. significantly less than folic acid, but some studies, depending on the type of folate, or if there's things like vitamin C that help absorption, or how much the person is eating, it can be as high as 60 to 90%. However, the validity of the approach used for these bioavailability studies is under question, and should we be comparing the absorption of food folates against a synthetic supplement? So that's a question. There's a reference there, and there are people that are starting to question this now and say, well, really, should we be saying that food folates are absorbed less efficiently when they're actually coming in an active form? Because it'll come in as tetrahydrofolate or 5-methyl tetrahydrofolate. It doesn't have to go through the dihydrofolate reductase. It was previously thought that folic acid, like our food folates, like our dietary folates, were converted in the small intestine into 5-methyl tetrahydrofolate. However, we now know that most of it is metabolised in the liver. So it's not actually metabolised in the same way as our food folates. Studies suggest that high-dose oral folate conversion is inefficient. This is because there is a limited capacity in the liver to actually convert folic acid. The magnitude of this is dependent on the dose and the frequency of the folic acid intake. So what I mean here is the amount of folic acid that's getting absorbed into the circulation, or this unmetabolized folic acid, depends on how much someone is actually consuming. So here we can see we're in the gut, we've got folic acid, we've got food folates, the polyglutamate form, that's with the N there. We've got our monoglutamate form, so the polyglutamate tails have been chopped off, and then we've got 5-methyl tetrahydrofolate down the bottom. So starting at the top, folic acid can be transported across that cell membrane, so it can be absorbed because it's in the monoglutamate form, but it needs to go through the enzymes dihydrofolate reductase, forming dihydrofolate, then through that same enzyme again to form the tetrahydrofolate. Then it can be used in that methylation cycle. When we have our food folates, we've already mentioned, they need to be converted to the monoglutamate form. This happens in the gut. And these can come in as the tetrahydrofolate, the 5,10-methylene, or even the 5-methyl. So when we eat food folates, we get the various natural active forms. And then 5-methyl, if you were having that as a supplement in the monoglutamate form, that obviously gets absorbed straight across that cell membrane and can go into the circulation. So what we're showing here too is that folic acid, though, being in the monoglutamate form, can actually bypass this particular metabolic pathway, particularly if these enzymes get saturated, and we'll discuss this, and we can get this unmetabolized folic acid in our circulation. So we have particular folate transporters that actually pick up the folate and help it get into the cell. So I've been mentioning that the monoglutamates can cross over the cell membrane, but I haven't mentioned these receptors that actually have to help bring it into the cell. So we have the reduced folate carrier, RFC1. For those of you who already do genetic testing, particularly those who do the direct-to-consumer, what you'll see when you're looking for this receptor or the folate carrier is SLC19A1. So it's usually written as SLC19A1. Anytime you see SLC, it's generally a transporter of some sort. So they're solute carriers. There's also the protein-coupled folate transporter, PCFT, and we've also got the folate receptor. We don't know as much about the folate receptor. We probably know the most about the reduced folate carrier, or RFC1. So these three systems have distinct tissue distributions. For example, the reduced folate carrier, I believe, is found in most of the cells in the body, so helping get folate into the cells, whereas PCFT is more so in the gut. And as I said, we don't know as much about the folate receptor, but what we do know is the folate receptor actually has a higher affinity, or it prefers to bind to folic acid, which is different to both the reduced folate carrier and the proton-coupled folate transporter. These actually have a higher affinity or prefer the food folates. So they will attach to those and bring them into the cell, over bringing in folic acid. However, there is some competition, and it depends on the dose of the supplements that someone is taking. So there are things that actually affect these transporters or these carrier proteins. There are factors that affect the upregulation, so the switching on of these carriers or transporters. If someone is folate deficient, there is an upregulation, so these transporters are switched on and people will absorb more of the folate. Also, vitamin D has been shown to upregulate or switch on these particular transporters. So think about vitamin D too if someone's folate deficient. There's also downregulation. So studies have shown that high-dose folic acid, and in particular this study here, there's an in vitro study that demonstrated a downregulation, so sort of turning down. There is less expression, less of these folate transporters or carriers being expressed in the intestine and renal epithelial cells cultured in particular cells that were supplemented with high folic acid. So really what it's saying is if you have lots of folic acid, you'll actually start to turn these things down, so you'll absorb less. And it makes sense. If you're low in folate, you're going to absorb more. If you're having lots of folate, we're going to switch these things off or down-regulate them. So think about that too. If you're giving your patients high-dose supplements, they're then going to be making adjustments because they don't need that much. It's not here, but I will tell you also that there are studies showing that people that do have long-term high doses of supplements such as folate, because of these down-regulation mechanisms, if they stop taking folate, they become folate deficient much faster than someone that wasn't supplementing because the body adjusts. So let's get into the important genes and the SNPs, the genetic variations. So I've mentioned dihydrofolate reductase a number of times. It's something that some of you may have seen if you're familiar with genetics. Not all genetic testing companies test for dihydrofolate reductase, and there is good reason for that, and that's because the evidence is quite controversial. We don't really know if the SNP, and it's not actually even a SNP, I'll talk about it in the next slide, it's more of a deletion, affects the enzyme. But some of you may be aware of it because the direct-to-consumer testing companies have it, but most of the other testing companies do not test for this. But I keep mentioning it, and it is important because it is the enzyme that metabolises folic acid. I still sometimes get people that think that MTHFR is what metabolises folic acid. It's definitely not. It's the dihydrofolate reductase. So the dihydrofolate reductase is required to convert folic acid into the functional form. The expression activity in the human liver is relatively low, and remember that's where the folic acid needs to be converted. Oral doses of folic acid between 260 and 400 micrograms, so sort of normal to low doses of folic acid, can saturate the liver's ability to convert folic acid, and this results in that unmetabolized folic acid as we've discussed. In addition, I talked about the regulation. If someone does start taking supplements, it actually turns on dihydrofolate reductase, and that makes sense. The enzyme is needed to convert it, but over time it can become saturated. The other thing I'd like to point out is that dihydrofolate reductase does not just convert folic acid or dihydrofolate into tetrahydrofolate. It's also involved in the biopterin pathway. So if someone's having a lot of folic acid, now this is theory, we don't have hard evidence for this, but it's something to consider because we do know, as you'll see in the talk, that folic acid can actually sort of block up dihydrofolate reductase. It's then not available for the biopterin pathway. So if you've got patients with mental health disorders, issues with serotonin neurotransmitters, you may want to consider that. So with dihydrofolate reductase, it's not actually a SNP. So we're not talking about a single nucleotide polymorphism, an A to a C or a T to a G. There's actually a 19 base pair deletion. So remember in the early modules I spoke about all the different types of genetic variations. We have SNPs, we have insertions and deletions, we've got copy number variants. In this case it's a 19 base pair deletion. You can see the RS number there. And we don't fully understand the function of this deletion. As I said, that's why not everyone tests it, because the evidence is not as strong as we'd like. But what we have seen is that people that inherit the deletion, so think of it as like the homozygous, one from mum, one from dad, there have been studies showing that there is more unmetabolized folic acid and lower red blood cell folate in individuals receiving supplements. So it does indicate that there probably is some functional effect. So if you do have the results for this particular gene and your patient comes to you and is asking about folic acid, I do think it's worth looking at. Even though the evidence is not as strong, it is the enzyme that's directly metabolizing this. So MTHFD1 is a very important gene. I've personally done research on this. It is really the enzyme that leads into that form of folate that's needed for DNA synthesis and repair. So it converts folates, or the tetrahydrofolate, into 5,10-methylene. There is a particular SNP, the 1958 G to A. So there's a couple of SNPs in MTHFD1, but this is the one by far that has the most research and that you want to look out for. Human studies have shown that there is a link with pregnancy complications, possibly neural tube defects. Actually, the reason why I've done research with this SNP is that paper around neural tube defects came out just before I started my PhD. However, they've followed this up with animal studies and they've found that it's not a cause of neural tube defects, but that it does decrease folate synthesis, disrupts the folate cycle, as well as the production of these nucleotides, and it can impair fetal growth, but not necessarily a strong cause for neural tube defects. But this SNP basically slows down enzyme activity, may increase the need for someone's folate, and thinking here too, you know, we're talking about folates that can support the DNA synthesis side of things, perhaps folinic acid, and there are some studies suggesting that people that have this SNP may even benefit from more choline. So people that have this SNP and have a low choline diet are at increased risk for choline deficiency. Remember, choline is also a methyl donor. So what you see in that methylation cycle is quite a simplified version, and that makes sense. Most of the time when you're looking at the methylation cycle, it depends on what the author or whoever's produced it is focusing on. You are getting simplified versions, and with MTHFD1, it's actually a trifunctional enzyme. And I'm going in a little deeper here to actually explain that because, you know, doing this module, I think you actually need to know really what the function of this enzyme is. This is not something that you'll then have to explain to your patient. I think this is probably getting a bit deep, but for you to truly understand what MTHFD1 or methylene tetrahydrofolate dehydrogenase is doing. So most enzymes like MTHFR actually just convert, you know, one form of folate to another. It has one function. Whereas MTHFD1 is a trifunctional enzyme in that it can do three things. It converts tetrahydrofolate into 10-for-mil tetrahydrofolate. It can then convert the 10-formyl tetrahydrofolate into 5,10-methenyl tetrahydrofolate. And if we're getting specific and technical and more accurate, it's actually folinic acid comes in here. And there's another enzyme, MTHFS, that converts folinic acid into the 5,10-methenyl, and then it gets converted to the 5,10-methylene. So that's what you see in that sort of overall methylation cycle. So the MTHFD1 converts the tetrahydrofolate to the 10-for-mil, then into the 5,10-methenyl, then to the 5,10-methylene, and it's the 5,10-methylene that then flows through that methylation cycle. So the other enzyme, MTHFR, I'm sure most of you are aware of this one. This is what converts folate, or that 5,10-methylene, into 5-methyl tetrahydrofolate. This is an important regulatory enzyme. So if the body decides it needs to make more nucleotides for DNA synthesis, it will take the methyl group from 5,10-methylene, and then what you have left is dihydrofolate, and it goes back around. But if someone has high homocysteine or they need to support methylation, MTHFR will be upregulated and it will pull that form of folate, 5,10-methylene, taking it away from the DNA synthesis side, supporting that homocysteine metabolism. So it really is pushing folate over towards homocysteine metabolism and directly supporting the formation of methionine and methylation. So we know that there are two SNPs with very, very good evidence that influence the activity of MTHFR. They're at position 677 and 1298. Both of these SNPs reduce enzyme activity, the 677 more so than the 1298. And that individuals that have these particular SNPs are likely to have higher homocysteine. They're at increased risk for higher homocysteine, lower folate levels in plasma and whole blood, and show less response to folic acid supplementation. What we mean here is if they do have supplements, it takes them longer to bring down those homocysteine levels or to get the folate levels up where you want them. So just focusing on the 677, it's the CC that is the normal, the wild type, that's associated with normal function of the enzyme. The CT, the heterozygous, one normal, one variant, or one wild type, I should say, and one variant, that is associated with a 35% reduction in enzyme activity. And then the MTHFR-TT, the homozygous, is associated with up to a 70% reduction in enzyme activity. And I've already mentioned this in previous modules. So it's a significant impact on the enzyme. These people are likely to have less folate, particularly the 5-methyl tetrahydrofolate, which then obviously influences that homocysteine methionine side of the pathway. But as I always say, even though MTHFR is very important, it impacts on the enzyme more than other SNPs that we've been talking about so far. And also it has a lot of evidence supporting these results. The reality is it's still only one gene, and you really do want to look at what's going on in the pathway to get as much of a complete picture as you can. So moving on to fortification. So folic acid must be converted into the active form so that it can enter that methylation cycle. Folic acid was being used as a supplement and had been found to alleviate folate deficiency anemia before we actually understood the methylation cycle. So folic acid has actually been used since the 1940s. It's been around for a very, very long time. And when we did learn about the methylation cycle or folate metabolism and realised that this particular form of folate isn't actually one of the active forms, there appeared to be little immediate reason to look beyond folic acid or to use a different supplement because it was shown to work in relation to anemia and there were no known side effects. Plus, it's also cheaper and easier to manufacture and it's very stable. But as we've mentioned, we now know that it's not active. It must be broken down and metabolised into the active form, and that unmetabolised folic acid can get into the plasma. And there is some concern that this may pose some health risks. Theoretically, folic acid could interfere with normal folate metabolism through the competition with the active forms for the transporters. So I already mentioned that those transporters have a higher affinity for the food folates, but if someone's not eating a lot of food folates, or perhaps they're overcooking their veggies and not getting a lot from the broccoli, but they are taking folate supplements, there's going to be this competition and it's going to bind with the folic acid. Also competition with the binding proteins and the folate-dependent enzymes such as dihydrofolate reductase. So this is clearly just to show you exactly what dihydrofolate reductase is doing. We mentioned at the beginning of the talk that there are 2 double bonds. You can see here in the blue it's highlighted. And what dihydrofolate reductase does is it breaks that first double bond. However, that's quite difficult. It requires a lot of energy and it's actually a slow reaction. So dihydrofolate reductase requires NAD, so you're thinking here niacin B3 to help support dihydrofolate reductase. And once that double bond is broken, we've got dihydrofolate, which is still not active. It needs to get to the tetrahydrofolate. That's what picks up those methyl groups. Think of the THF. The same enzyme, using B3 again, then breaks that second double bond and then we have our active form. But because that first step is a lot slower, the enzyme can kind of get stuck there. So when we talk about saturation, that's what we're talking about, it becoming saturated and it's trying to convert that folic acid down to dihydrofolate. And because dihydrofolate and folic acid are not active, that can actually end up representing like a functional folate deficiency because the folate's stuck here and not getting into the actual methylation cycle. So I've written it here, high folic acid can competitively inhibit the conversion of dihydrofolate to tetrahydrofolate, which is what we need, the active form. And because they're not metabolically active, this could create an intracellular folate deficiency. Again, a lot of this is theory. I want you to know there is not hard evidence for this, but these are concerns that practitioners are asking, patients are asking, and now that we're really understanding these metabolic pathways, we do have to keep open-minded and question these things. The other concern that is more well known with folic acid is that it can actually mask B12 deficiency. So if folic acid does mask B12 deficiency, this can lead to a delayed diagnosis and possibly the progression of neurological damage. And sometimes this neurological damage is irreversible. What I say to all of my clients and patients and practitioners, and even when we were doing research, is not to give folate without the B vitamins. When you think about that pathway, we know that you need B12. If you're giving folate and you want it to go and convert homocysteine to methionine because someone comes in with high homocysteine, if they're low in B12, you get what's called the folate trap. It gets stuck. So you need to ensure there's enough B12 so that folate can move through to the homocysteine side. If you're giving folic acid and you want it to come down for DNA synthesis or through MTHFR, you need your B6, your B2. So you need to think about the other Bs as well. So generally, I would suggest, particularly for those with genetic variations and SNPs, meaning the enzymes are slower and we need support. So for example, with MTHFR, studies have shown that giving B2 riboflavin, which is the cofactor, does increase enzyme activity. That is the purpose, to give these cofactors to try to sort of compensate for these genetic variations. Consider a B complex. However, we know that not all of our patients can tolerate a B complex. Now, I know this is a very small proportion of the population, but many of the patients and clients that we tend to get are the complicated ones that are sensitive. So if you do want to give one supplement at a time, because that's what that patient needs, you want to be thinking about your B12 and your cofactors, and they have plenty of those so that the folate isn't getting stuck or isn't causing an upregulation or a downregulation of transporters, and ultimately perhaps even causing a functional folate deficiency because it's getting stuck somewhere in the pathway. You know, you're actually causing more of a problem if you start to give folate and it gets stuck somewhere. So the other thing that needs to be addressed is folic acid and cancer. This is another concern from many, many people. There has been studies for a very, very long time showing that folic acid can actually increase the growth of cancer. So if someone already has cancer and you take folate, you're actually helping the body to make these nucleotides, what is needed for DNA synthesis. So you could exacerbate the growth of that cancer. There's no doubt that giving folate to someone with cancer is likely to not be a good thing. I mean, maybe a little bit because you want them to get well, but particularly if you're getting to high dose, that can be a concern. I don't necessarily think that folic acid or high folate causes cancer, but it definitely can stimulate the growth of these cancers, and there is a lot of evidence to show that. There are some studies, there's only 2 that I know of, and both of the references are there, suggesting that supplementation with high-dose folate or folic acid may contribute to immune dysfunction, in particular affecting the natural killer cells. That is a concern. There's not a lot of evidence, but there are a few papers. But I also want to remind people that folate deficiency has been linked to cancer susceptibility because of increased DNA damage. So if you're low in folate and you're not being able to make these nucleotides, you actually get more DNA damage, and that is one of the underlying and major causes of cancer. So you want everyone to have adequate folate, but consider, if your patient has cancer, not giving them folate, in particular folic acid. That's where sort of the studies lie. But I believe when you think about the function of folate, whether it was 5-methyl or folinic acid could be a concern too, because you're still going to be feeding that cancer. So folic acid exposure. So high folic acid intake is common because of fortified foods, flour, breakfast cereals, breads, and some people taking vitamin supplements. The recommended dietary intake for folate is 400 micrograms or 600 micrograms in pregnancy. And we know that higher doses, up to 5 milligrams a day, or particularly 5 milligrams a day, are recommended for pregnant women with certain preconditions, such as obesity, diabetes, in some states, in South Australia where I was, for those that have the MTHFR genetic variations, and also the history of neural tube defects. So when we talk about folic acid fortification, cereals are generally fortified with about 100 to 400 micrograms of folic acid per 100 grams. I read the back of everything. I'm sure many of you also do that. I would say most of the time it's closer to that 100 micrograms. And when you consider a serve, which is recommended depending on the type of cereal you're eating, they say that a serve is about 40 grams. It's not too much. However, I don't really know anyone that eats 40 grams of cereal. You know, some people have huge bowls of cereal. So you're actually having quite a high intake of the folic acid if you are consuming these processed cereals. In addition, it's in our bread. So if someone's having cereal and they're having a few sandwiches, they could be having quite a high intake of folic acid. And you need to consider these on top of, the dietary intake, what you're getting from your foods, the natural forms of folate, and what this could be doing to dihydrofolate reductase that has a limited capacity in the liver. And we know that the folic acid has to be converted in the liver into these active forms. So both folate deficiency as well as excessive dietary folates, really this should be more so folic acid, because the study's on folic acid rather than dietary folates. Compromised red blood cell formation, resulting in defective cell cycle progression, persistent DNA damage, and impaired production of lymphocytes, so affecting the white blood cells and the red blood cells. This information comes from the study that I've got there, and I've heard people talk about this. So as I said, there's these theories around folic acid and high-dose folate causing functional folate deficiencies, and it actually makes sense. We do know there's this folate trap. But when you look at one of the papers that I've been told about that actually provides some evidence for this, and it's in a mouse model study, obviously we couldn't do this in humans, this study is really not a good comparison with humans, because actually this mouse model study, they gave 10 milligrams per kilogram of folic acid. So let's just think the average person is 60 to 70 kilograms. That's 600 to 700 milligrams of folic acid. Now, as I mentioned before, the high dose that we frequently give is 5 milligrams. I know some people occasionally go higher, but when I'm saying high dose, in my mind and some of these other papers, we're more thinking more than 1 milligram. So this is really, really high, and I am not surprised to see there are issues here, because we know anything that you have in excess is going to cause some kind of negative health outcomes. So just be cautious when you're reading the papers and actually have a look through the methodology or what doses people are talking about, because this is definitely supraphysiological levels. I also mentioned they're sterilised by irradiation. We can't really account for that, and I don't know if it's going to affect the food, the mouse chow they were eaten. But remember, well, and I say it in the coming slides, folate is actually broken down with heat and light. So I'm wondering if it was irradiated, that would actually bring the levels down. But it's still excessively high and not a direct comparison that we could use for humans. The upper limit for folate, or for folic acid, is 1,000 micrograms, so 1 milligram. It's thought that 400 micrograms of folic acid in addition to dietary intake is safe, and there's no upper limit for natural folates. So what are the pros and cons for fortification? So the proponents argue that fortification should be mandatory because supplementation and dietary modification are largely ineffective in preventing neural tube defects. 50% of pregnancies are unplanned. So 50% of pregnancies are unplanned, and people are not finding out until after that first month. They're really missing that window to reduce the risk of neural tube defects. So that is the main reason for fortification, and we will talk about this in a bit more detail in Module 5 in Hormone and Fertility. It's also suggested that folic acid may reduce the rates of cardiovascular disease and cognitive decline. I've not seen the data for this. All of these outcomes are measured. There are statistics, so hopefully we'll start to see some indication since the fortification of folic acid with these kind of outcomes. The opponents contend that health benefits are outweighed by the theoretical risks of adverse effects, so neuropathies due to the masking of B12, as well as the possible cancer risk. At present, expert groups have stated there is no conclusive evidence that exposures to unmetabolised folic acid causes adverse health effects. What they're really saying is we need some more hard evidence before we change this fortification program. Currently, folic acid is the first choice for supplementation or fortification because of its stability and low cost to produce. However, what I want to point out, and I think many of the practitioners in this audience would know, is that we now have natural, active, commercially available and stable supplements with comparable bioavailability that are physiologically active and have the ability to improve folate status at similar doses than folic acid. And these are the 5-methyl tetrahydrofolate and the folinic acid, or 5-formyl tetrahydrofolate. And we know that we can now get the 5-methyl tetrahydrofolate in that calcium salt. It's now stable. It can be used. So we have gone through the folate pathway. We've talked about dihydrofolate reductase. We've talked about MTHFD1 and MTHFR. I haven't focused on SHMT, serine hydroxymethyltransferase. This also converts tetrahydrofolate to that 5,10-methylene tetrahydrofolate. There is a genetic variation that can slow that down. So these are sort of the main genes and enzymes that you want to look out for in regards to folate metabolism. And hopefully now, or you may have to go back and watch the module again, you're very clear on knowing exactly how folic acid enters the pathway with folinic acid. Remember, this is quite simplified. I showed you that MTHFD1 where the folinic acid really comes in, and the 5-methyl that many of you are familiar with. So that 5-methyl tetrahydrofolate has to donate its methyl group onto homocysteine, which we'll move on to form methionine. So the homocysteine methionine pathway has a number of genes with genetic variations that can influence homocysteine metabolism and the production of these methyl groups, or S-adenosyl methionine, that provides these methyl groups. So MTR, or methionine synthase, is the enzyme that picks up that methyl group. It requires methyl B12, and it's the MTRR that works with that tries to ensure B12 is active so it can pick up that methyl group. Zinc is also important. So for MTR to hold on to homocysteine, it requires zinc. So think about zinc as well when you're trying to support homocysteine metabolism. And we know that methionine, which is this essential amino acid, is needed to make SAMe, and it's SAMe that provides all of these methyl groups, absolutely essential for methylation throughout the body. Homocysteine is a non-protein amino acid. What I mean here is it's an amino acid that doesn't actually make proteins. It's like an intermediate. So homocysteine is important for making methionine, which is an essential amino acid, but also it can go down the transsulfuration pathway to form cysteine, or cystathionine and then cysteine. High levels have been associated with various health disorders, cardiovascular disease, neurological conditions, pregnancy complications, and the optimal range is likely to be somewhere between 5 and 9. So studies in relation to cardiovascular disease definitely suggest under 9 to prevent cardiovascular disease. Dale Bredesen recommends 5 to 7 for reducing risk of Alzheimer's. So these are people that already have early cognitive decline, so that's much lower end. And then research that I have done, I would say between 6 to 9 for reducing risk of pregnancy complications. Now know that when I say 6 to 9, it does actually depend when you are taking the homocysteine levels. So homocysteine actually drops significantly in pregnancy. By the time you get to 20 weeks gestation, it comes down significantly. So if you are measuring it before pregnancy or early stage pregnancy, no, it's going to come down. So a homocysteine of say 6 pre-pregnancy possibly could be a bit low because you need to have enough to make your methionine and your cysteine. But during pregnancy, that's a good level. And high homocysteine or homocysteine levels are heavily influenced by our B vitamins, the folate, the B12, the B6, and the genetic variations. So methionine synthase, also known as MTR or MS. So it's generally shown as MTR, and that's because the actual name is methylene tetrahydrofolate homocysteine S-methyltransferase, a huge mouthful. Hence we say methionine synthase. But it is actually MTR. Occasionally you'll see it as MS, but just know they're all the same thing. It converts homocysteine to methionine using a methyl group from 5-methyl tetrahydrofolate. B12 is the cofactor. I mentioned that it needs zinc to hold on to that homocysteine molecule. And the most studied SNP is that A2756 to G. And the research is conflicting. For those of you who look at genetic test results, you may see that one genetic testing company shows the A as red, another one shows the green as red. This is one of those ones that you can see conflicting information, and that is because depending on the research you read, sometimes it seems that the A might be a bit faster or a bit slower. I think the issue here is because B12 is the cofactor, which is essential to drive that enzyme activity. If someone's deficient in B12, the enzyme won't work. It's more to do with the B12 actually driving this enzyme than the SNP. So I think the SNP actually only has a small effect on the enzyme activity. When you put all of the research together, it does look like it's the G allele that increases activity, but just know that it's a little conflicting. And if you are comparing results, you will see that some companies report it differently. Focus on the B12 levels, but it's still worth mentioning, and I'm telling you because people ask about it. All the genetic testing companies test for it, and it is absolutely essential in that homocysteine pathway. But the evidence for this one is not as good as some of the others. Methionine synthase reductase, this regenerates oxidized B12. Oxidized B12 cannot function, it can't bind to MTR. So MTRR reduces it back to the active form so that it can pick up that methyl group and help support methionine synthase. The SNP in MTRR has more evidence or is a little more conclusive than the MTR. We know that the 66G, so A66G, is the SNP with the most evidence, and I would suggest for those of you, some companies test multiple MTRR SNPs, focus on this one. The G allele is associated with higher homocysteine levels. And again, the mechanism is not completely understood, but we think it's thought to affect the binding of that B12 complex. So MTR, MTRR, B12, homocysteine, they all come together to catalyse that reaction of homocysteine to methionine. And that's really showing that here. We can see the MTRR, the cofactor is B2. Some studies suggest B3 as well, but generally FAD, so B2. And what it's doing is regenerating this oxidised cobalamin so that it can pick up that methyl group and support the function of MTR. Another one to mention that helps to convert and regulate our homocysteine levels is BHMT, betaine homocysteine methyltransferase. So betaine homocysteine methyltransferase is predominantly found in the liver and the kidney. It converts about 50% of the homocysteine in the liver to methionine, so possibly very important for detoxification. It requires betaine, which can be oxidised from choline, and the SNP, again, it's not nearly as evidence-based as, say, MTHFR, but there is enough evidence to show that this particular SNP is associated with increased homocysteine and lower dimethylglycine, which you'll see in this image is actually the product. So the SNP actually results in higher homocysteine and lower dimethylglycine, suggesting that it actually affects the enzyme. It's slower. So if someone's got MTR, MTRR, BHMT, even CBS that we'll talk about in the next module, really their risk for high homocysteine is much higher. So finally, moving on to B12, this is really just a simplified diagram to show you two of the genes that you want to be looking at in relation to B12, so FUT2 and TCN2. So fucosyl transferase 2 transfers a fucose molecule, so a sugar group, onto the cell surface of glycoproteins and glycolipids. So FUT2 is predominantly expressed in the epithelial cells and in the tissues that secrete mucus, including the mucosal lining in the gut. It creates oligosaccharides. One of these oligosaccharides is involved in producing the H antigen. This is essential to synthesize our ABO blood type antigens. These oligosaccharides also act as a food source and adhesion structures so that bacteria can actually stick on the cell surface in the gut lining, and this affects the intestinal microbiota. So FUT2 does impact a lot on gut health and the immune system, but we will focus on that part when we get to the immune module. What we're going to focus on here is the association with B12. So there are a number of genome-wide studies, very, very strong evidence linking a number of SNPs, but in particular, this is the one you want to look out for, with B12 levels. This RS601338 is the particular SNP that we associate with secretor status. What I mean here is we talk about the secretions of those ABO blood type antigens. I just said the FUT2 has to transfer that sugar molecule that helps make the blood type antigens in these mucosal linings. So if people don't secrete their ABO blood type, they're known as a non-secretor. Non-secretors are the AA genotype. The AA genotype actually results in a non-functioning protein. So as long as you've got that G allele, if you're GG or AG, you're going to be a secretor. What that means is that you're going to be able to transfer that sugar molecule, you're going to make the oligosaccharides that act as a prebiotic, you're going to make that H antigen, which helps make those blood type antigens. There is a strong association with FUT2 and serum vitamin B12 levels, as well as homocysteine levels. So interestingly, it's the secretors, which actually generally have better gut bacteria, that are lower in B12. But it's a bit more complicated. Even though they have better gut bacteria, and what I'm talking about here is bifidobacterium and lactobacilli, they also are more likely to have pathogens such as H. pylori. Because they've got a better gut environment, they've got these adhesion molecules, things like H. pylori can come along and stick. So these people are at increased risk and more likely to have H. pylori. And we think, we're not sure, that is the mechanism relating to B12, because we know that those that have H. pylori, it's about 50% of the population, have much lower B12. So we think it's affecting the gastrointestinal absorption. So we don't completely understand the mechanism. It's highly likely due to absorption, but we definitely know there is a strong link, and it's been replicated multiple times in genome-wide studies with B12 levels. The last one to talk about, transcobalamin 2. So this is a vitamin B12 transport protein. It helps get B12 to target tissues and then across that cell membrane. There is a particular SNP, the C776G. The G allele is associated with reduced B12 levels compared to that CC or the wild type. So the GG is associated with increased homocysteine as well, and vitamin B12 associated disorders. So if you're concerned about B12, you want to be thinking about FUT2. Are they likely to have lower levels? And are they getting it into the cells efficiently? So just quickly, I mentioned that we'll talk more about this in the neurotransmitter module, but there are various forms of B12 supplements. You know, I went into detail about the folate supplements, but with B12, we've got cyanocobalamin. This is the most common form. It's not active, contains cyanide. It's considered non-toxic, but it must be eliminated. It still needs to be detoxed from the body. There is hydroxycobalamin. This is found in our food. It's natural, but it still must be converted to those active forms. And the active forms are the adenosylcobalamin and the methylcobalamin. So there is actually a paper. So with the folates, really we're talking about absorption and theories and trying to understand what may be the best form of folate. But in relation to B12, we actually have a paper that has looked at the best type of B12. And what it suggests is that if someone is B12 deficient, that actually methylcobalamin and adenosylcobalamin at 500 to 750 micrograms really is the best way to address B12 deficiency, and it's addressing both functions, adenosylwith the mitochondria and the energy, methyl with the methylation cycle. It says that if someone is a smoker because they get cyanide, high levels of cyanide, that hydroxycobalamin is better. And I would suggest for your patients, if someone does have high levels of toxins or chemicals, thinking about possibly hydroxycobalamin. The paper also concludes that the oral route is comparable to the intramuscular route for rectifying B12 deficiency. Getting to the end, we know that B vitamins are water soluble and they're found in many foods, but they're easily destroyed. So I've talked a lot about supplements, but we know we need to educate our patients about the food they eat. Food processing reduces the amount of B group vitamins, making them less nutritious than whole grains. Think about people eating white flours and white rice. Teach them about whole grains if they can tolerate grains. The body has a limited capacity to store most of the B group vitamins aside from B12 and folate. So if someone has a poor diet for just a few months, they may ultimately be getting quite low in B2 and B6. And just clinically, I actually am seeing a lot of low B2 levels these days. It's important that adequate amounts of these vitamins are consumed regularly to ensure that people do not have low levels. So what are the symptoms? So there are a wide variety of symptoms associated with B12 deficiency. This ranges from fatigue, weakness, anemia, skin rashes, neurological side effects as well, or disorders. And we know that genetic variations within key genes coding for enzymes in the methylation pathway increase risk for lower B vitamins. So if someone comes into you and they've got these SNPs, you need to think, do they need more folate? Do they need more Bs? Are they really tired? Do I have to support, methyl B12? Various testing can be done and will be discussed in the case study, because even though we've got the genes and they increase risk, we actually need to know levels, and we will talk about this when we get to the case study. So what can go wrong if someone's got these genetic variations, if they've got a low intake of B vitamins? We know it can influence fertility, in particular infertility and pregnancy complications, cardiovascular health, mental health disorders, immune conditions and hormonal conditions. So this is why we hear so much about methylation, because it can be an underlying cause and trigger in many, many health disorders. Thinking about the diet, we need to think about foods that are high in folate and B vitamins. With the folic acid, we know it converts dihydrofolate to tetrahydrofolate. It requires dihydrofolate reductase that is saturated at low levels So if you are going to give folic acid, and I'm not saying don't, because the evidence sort of under that 400 micrograms isn't too bad. A little bit of folic acid is likely to get metabolised and used efficiently. It's when we get to the high doses that we start to see issues. Folinic acid is what converts 5,10-methylene. Tetrahydrofolate directly supporting DNA synthesis and repair. Consider those SNPs, MTHFD1 or SHMT1. The 5-methyl, this bypasses MTHFR. So if someone's got multiple SNPs or you're concerned about MTHFR, you can think about the 5-methyl. We know that we have hydroxy and cyanocobalamin, but these must be converted to the active forms. Think about the hydroxy with chemicals and toxins. Adenosine, which supports energy, and methyl, which directly supports methylation, the MTR and the MTRR. Again, I've said it throughout the talk, also the cofactors, your B6, your B2, vitamin C can help with folate absorption and other methyl donors. If someone's got high homocysteine and you're giving them folate and B vitamins, most of the time the homocysteine will come down. But I have seen it occasionally where homocysteine doesn't come down. Think about choline, think about other methyl donors. Perhaps it's BHMT, something's going on in the liver. So these are a resource for you. I'll touch on these in the case study. This sort of summarises a lot of the genes. Just want you to know that when you are looking through them, this is not based on a meta-analysis or a systematic review. This is my opinion looking at all of the literature, but it's a resource for you to understand these SNPs. I'm also not saying everyone has to have those supplements, but they're the supplements that interact with those genes. So our take-home messages are that balanced methylation is essential for optimal health and is influenced by nutrient intake and inherited genetic variations. Nutrigenomics helps to identify genetic variations that influence folate, as well as our B vitamins and homocysteine levels, and this can guide our dietary advice as well as the supplements we want to prescribe. Methylation markers, including things like homocysteine, can be corrected and balanced with optimal nutrient intake. With folate, generally a maximum of 1 milligram. Of course, this is personalised medicine. Some people need different amounts. These are just general guidelines. But really, 1 milligram, unless they've got neural tube defects, a BMI over 30. They must have adequate cofactors. Majority of the evidence actually still supports folic acid, but the lower levels are probably more efficient because of this saturation effect with dihydrofolate reductase. Emerging evidence suggests that folic acid may not be the best form, particularly at high doses, and those with folate-related SNPs, that slow enzyme activity may benefit from these active forms to bypass all of these enzymes and help support methylation. Thank you.